SRP-9001 is being evaluated in a comprehensive clinical development program. There are 4 studies that are being conducted to date, but with plans to evaluate in a very broad patient population. Sarepta is committed to getting not only this therapy, but all therapies. Our goal is to treat 100% of Duchenne patients. The 4 studies that I mentioned include Study 101, Study 102, Study 103, which is also known as Endeavor, as well as a phase 3 study known as Embark, or also 301. That phase 3 study is a randomized placebo-controlled study that is fully enrolled. We've dosed 126 patients as of September 2022. Safety, protein expression, and functional outcomes are available for those first 3 studies that I mentioned. So again, Study 101, Study 102, and specifically Cohort 1 from Study 103, as well as there's data available for an integrated analysis of all 3 of those studies at the 1-year time point. And so across all of those studies, we've seen robust SRP-9001 protein expression. And the way that we assess that is through muscle biopsies. Those muscle biopsies are taken 12 weeks after that single IV gene therapy dose administration. And we measure protein expression by Western blot. And then in terms of biological endpoints, there is other valuable information that we get from those biopsies, including we were able to confirm that the protein is, the SRP-9001 protein is present at the sarcolemma, which is where it needs to be in order to do its function, which is stabilize the muscle cell membrane. And we also determine vector genome copies, which really confirms effective transduction. In other words, delivering the gene to the target cells. So we've seen consistent results across all of those studies for the biological endpoints, also seen consistent studies across the studies around functional endpoints. So we've seen improvements in physical function. And the primary endpoint that we evaluate for physical function is NSAA, or the North Star Ambulatory Assessment. This is a 17-item scale. It's a validated tool that is used to assess motor function in ambulant individuals with Duchenne. And each of those 17 items is scored from 0 to 2. And so a zero indicates that the individual cannot perform the activity. A 1 indicates that the individual can perform the activity with modification. And a 2 indicates that that individual can perform the activity normally. So the maximum score on that NSAA scale is 34, and that will help put into context as I give a summary of the clinical data. I do mention that NSAA is a primary endpoint that we evaluate for functional outcomes. We also look at time function tests, such as time to rise, as well as the 10-meter walk run. But again, that NSAA is the primary functional endpoint from the studies. And what we've seen, I'll start with Study 103. So this is a study in patients between the ages of 4 and 7, ambulant patients. And this is particularly specific to Cohort 1. There are a number of cohorts in that study, but for Cohort 1, what we've seen at 1 year is a mean difference between SRP-9001 treated patients and a propensity-weighted external control cohort of about 4%. I'm sorry, 4 points on that NSAA score. And so really what that means, about one of the common questions that we get is, what is that clinically meaningful? And so when you think about that NSAA scale, what that means is that an individual could potentially now perform 4 of those activities normally where that required modification before. Or it could, another example would be that the individual can now perform 2 of those activities that they could not perform at all. And so that really gives you some context as to the clinical meaningfulness of those NSAA improvements. With Study 102, the most recent data that we reported was for 2 years of patients that were treated in part 1 of the study. This was the first double-blind randomized placebo-controlled study, and we now have up to 2 years of data, and that those first patients that were treated with SRP-9001, and again showed clinical improvement, statistically significant treatment difference with respect to the NSAA, a median difference of 5 points. So now seeing those clinical changes or clinical improvements at a 2-year time point. And then for study 101, this was our proof of concept study, early study of 4 patients, again, an open label study that we now have up to 4 years of data on. And for those boys, again, looking at patients between the ages of 4 and 7, an ambulant patient population, we saw a mean change or treatment difference in the NSAA of 10 points when compared to the external control cohort, which is a propensity weighted scored external control. And the significance of that 101 data and that 4-year data that we have to date is it really confirms the durability of that clinical endpoint. The studies to date that we have conducted are in an ambulant patient population between the ages of 4 and 7. But here, 4 years later, these boys in the 101 study are about 9 years of age. And what we know from natural history is that these boys would be predicted to be in a steep decline phase, and yet we're seeing this 10-point difference on the NSAA. So we're very encouraged by these results and really excited about the potential opportunity of bringing this treatment to boys that need this therapy in the near future.